So what I want to do today is to talk a little bit about the other side of the gene drive coin. So Andrea talked about suppression. Today what I want to talk about is this idea of population replacement, population modification. And so of course there are two components to this. One is spreading a transgene into a population, and the other is what do you spread in. So Tony James, who's going to be here on the panel, will be able to talk a lot more about the kinds of disease genes that he and others are trying to put into mosquitoes. Today I'm going to focus just on the question of how we get things into a population. And so this is the challenge. How do you spread a trait into a population? And as Andrea mentioned, the bottom line problem is that genes that you want to put into a population don't confer a fitness benefit. At best, they're neutral. Usually, they have some sort of cost in terms of life history. And so the goal of all of our work, and that of a lot of other people, is to create something called a gene drive element that has, that when it inserts into a chromosome, and we're going to talk here about elements that remain at a fixed position that happens to have a cargo gene linked to it, and that's going to do this magical thing where the gene drive element is going to reverse this fitness relationship such that now the gene drive carrying individuals are more fit than the non-gene drive carrying individuals. So that seems a bit like magic. And seems like it shouldn't be possible. But what I'm going to tell you is that this is all just a series of genetic tricks that in fact nature uses all the time. And if we go back, the basic idea behind the genetic trick is, for example, let's say Fyodor and I are in line for a dessert, and there's only one left, and he's ahead of me. There are two ways I can get to the front of the line. One is I can be faster than him. I can be more fit. The other is I can just trip him. And this is what this kind of gene drive does. It's all about hurting the other as a way of getting ahead. So you can also think of it as a form of spite, sort of everybody's favorite emotion. Kind of. All right. Now, the important characteristics of a gene drive for replacement are listed here. So it needs to be stable in a population for a long time, needs to critically be portable to diverse species, extensible for multiple cycles of population modification. I'll come back to that. That's really the key point I want to leave you with. And then it should be, in some sense, reversible. And so over the years, we've actually, much to my dismay, been working on this for a long, long time, since 2007, when we published the first synthetic selfish genetic element, Medea. And then over the years, on our own and in collaboration with Tony, in collaboration with Dan Wattendorf when he was at DARPA, we've built a bunch of other elements that all can do population modification replacement in Drosophila in some way. And they, to some extent, have the characteristics that we'd like. But we've always felt that they fell short at some level, particularly in terms of portability to diverse species. Basically, the ease with which it was possible to make these things. Short answer is they took a lot of work. And so what I want to talk about today is a new approach we've developed based on CRISPR that we think will be much more useful for this sort of experiment in a diversity of species. And the two key people here are Tobin Ivey, a graduate student, and Georg Oberhofer, a postdoc in the lab. And so we're going to start off with Cas9, and the only thing we care about with Cas9 is that it cleaves. And so what we're looking at here is a germline promoter driving the expression of Cas9, 4 guide RNAs that create a multiplex that can then cleave an essential gene, and the key is an essential gene for either viability or fertility located anywhere else in the genome. That's all that matters. Now, when that happens, of course, Cas9 does what it does best, which is to cleave DNA. And in our case, we want non-homologous end joining. We just want to screw the gene up in some ill-defined way. We don't care how it gets screwed up. We simply want it to be a loss of function allele. Now, that's great. But the other thing we know we can do with Cas9, of course, is because it's all sequence-based recognition and cleavage, we can also create resistant alleles. But in our case, as opposed to Andrea's case, resistant alleles are a good thing because this is simply a recoded version of the essential gene that is now unable to be cleaved. And when you put these two things together, just two simple components, Cas9 targeting an essential gene and a recoded version of the essential gene, and you stick them at the same site in the genome, you have built a powerful selfish genetic element that will spread itself through a population. And so what happens here, of course, is that Cas9 is going to cleave this wild-type copy, and in doing that, it creates loss of function alleles. And the important thing is that this cell, this organism, is still alive because it carries a recoded copy of the essential gene. And just to bring us all back to the first lecture of genetics, third grade, this is because most genes, and we'll talk about exceptions in a minute, one functional copy is sufficient to give you the wild type, the normal phenotype. All right? So with those three ideas, cleavage, rescue, and one copy is sufficient, we get to do something interesting. So again, all of the interesting stuff occurs in the germline. For an organism that has a germline, interesting opportunities arise for organisms that don't have a germline as well. But the bottom line is you get cleavage, germline cleavage. Let's say this is a female. So the two wild-type copies are cleaved. The cell is alive because it carries a recoded copy. Those germ cells become an oocyte that goes through meiosis. One copy, one gamete carries the cleaver element, and one of them doesn't. Now, very importantly, Cas9 guide RNAs are stable as protein RNA complexes, and thus they get carried over into the embryos that arise from these oocytes. And what that means then is that when this female, heterozygous female, mates with a wild-type male, all of his wild-type copies of the essential gene get cleaved, and thus the only individuals that survive are those that inherit the cleaver element. Those that fail to inherit cleaver die because they inherit no wild-type copies of the functional gene, no functional copies of the essential gene. Now, this works in animals because gametes don't do transcriptional work, right? The transcription occurs in the diploid stage, and then the components are simply shared with the haploid stages. But there are a lot of organisms like yeasts and plants where gametogenesis actually requires a lot of gene expression during the haploid stage. And if you just sort of walk through the logic of this cleaver element, you'll see that again, as spores are created or pollen like gametes are created, the ones that will survive and be able to fertilize are those that carry the cleaver element, whereas those that won't because they lack functional copies of an essential gene that, say, is required for pollen tube growth, spore formation, et cetera, et cetera. Okay, so all of this brings us back to this idea of how do you increase the fitness of something that's inherently unfit or less fit than a wild-type organism. And in this case, it's literally by creating a genetic condition that results in the death of those that don't carry the selfish genetic element. Now, the population biology of this is nice. It is what it is. And what I mean by that is that when you introduce it at low frequency, illustrated here, the release ratio, and there's low fitness cost, it takes forever to spread because think about it. You have 1 in a million of these individuals. There's only one killing event that happens, and so there's a very little increase in the frequency. But the more you add, the faster it goes, such that when you add decent fractions of a population as transgene-bearing individuals, within about 10 generations, 6 to 10 generations, it will spread to genotype fixation. Now, importantly, it can also carry a fitness cost. So if there's a fitness cost, then you need to work harder in order to get it to spread. You need to add more to overcome this initial fitness cost to drive the fitness of wild type down below the fitness of the transgene-bearing individuals, but you can do it. You simply have to add more. But of course, it's not magic, and so if you're in this regime over here, where the fitness costs are large and the introduction rate is low, then you're still going to end up in this regime in which the wild-type individuals are more fit. Okay? Now, one final point before I get to a tiny bit of data, and that is let's imagine that the gene you're targeting is actually haploinsufficient or even haplolethal. Let's violate that initial condition I was mentioning. And it turns out that doesn't actually really matter very much. What it does is it shifts the curve so that you need to work harder in order to get an initial introduction frequency that crosses this threshold needed for drive. But that actually turns out, but then once you do that, it can go quite quickly in this region here, which is all this region here, less than 20 generations. So this actually turns out to be a useful feature in a way, because what it does is it creates what's called a threshold-dependent drive, such that if there's some level of haploinsufficiency for the loss of the gene, then you have to add a significant fraction of the total population in order for drive to occur at all. And so if you're thinking about accidental releases or movement from a target area to a non-target area, this actually can be a useful feature because it's baked in to the genetics of the drive system. It's unlikely that you will change the haploinsufficiency of a gene through genetic modifiers at other loci. Okay. Now, one final point is everything I've talked about, of course, here has been and is, because this is what we've done with Cas9, but it's useful to realize, and I oftentimes like to think about, could I have done these experiments when I was a postdoc or a starting assistant professor? And the answer is yes, because of course, one could have done all of this with zinc fingers or TALENs, though you would have had the problem of multiplexing, that you need to avoid the creation of these resistant alleles. But the bottom line is that all of this is a 20-year-old idea, more or less. Now, as was also pointed out, the only thing we really care about here is DNA sequence modification. And so that can be a double-strand break, but it can also be NICs followed by base editing. It can also be NICs followed by the use of the prime system, a reverse transcriptase, or even non-RNA-based systems like site-specific recombinases, which have recently been engineered to excise particular regions of the human genome. So the basic idea is agnostic as to how the DNA modification occurs. All right, finally a little data. This is an experiment where we took a random essential gene and that is involved in mitochondrial ribosomal RNA function, and we introduced cleaver-bearing individuals into the population at 25% allele frequency, which ends up with 50% of the individuals carrying the transgene to begin with. And what you see is that it spreads very quickly to the population to genotype fixation and has stayed there for about a year at this point. The controls in this experiment are just taking the rescue alone component, the recoded version of the essential gene, and just adding that to a wild type population and just asking if increasing the copy number of an essential gene does anything interesting. And what you see is that the interesting thing that it does is that it probably has a bit of a fitness cost. And then the dotted line is a model for what we would expect for an element with no fitness cost. And the data fits pretty closely to that. Now, we've also done this with two other genes. This is a gene involved in nuclear transcription, TF2A something, and this is a gene called dribble, has something to do with cytoplasmic ribosome biogenesis. And the important point I want to emphasize here is that we didn't read a single paper on these genes. I have no idea what they actually do. I mean, I do a little bit, but you don't need to know. The only thing you need to know is that they're essential, highly conserved, very few sequence polymorphisms, because they're really deeply, deeply conserved throughout all of animal evolution, all of eukaryotic evolution. Now, a final point I want to make about this is that once you end up in this regime over here, where you've gone to genotype fixation, what that means is you've cleaved all the wild type alleles in the population. And what that means is that literally now your population is absolutely dependent on, as in addicted to, the presence of this selfish genetic element. So it cannot lose that element at this point. And that makes it somewhat different than other drive mechanisms that we've worked on and that nature uses. All right, so that's the good news. But now let me tell you the truth. And the truth is that when you think about population replacement modification, you need to face up to the facts. And the facts are that it's gonna fail. It's gonna fail because everything fails when it involves interventions that involve mutation, selection over time. And we see this with cancer therapeutics, antibiotics, antimalarials, insecticides, herbicides. There will always be evolved resistance. And what that means is that you as a community will always be going through cycles of success, followed by failure, followed by retooling and going in again and trying to recapture the success that you had initially. And then, of course, in addition, any gene drive component, particularly the cargo, has the possibility of mutating to a loss of function phenotype. I mean, it's not a possibility, it's an inevitability. And so that will happen. And what that means is that we need to be able, and then in addition, the other thing to point out is that when you bought a phone 20 years ago, that wasn't the phone you want to have now. And so we always need upgrades, knowledge always increases, and we'll always want to add new functionalities, particularly if a vector, for example, now becomes a host for a new disease, like Zika with Aedes aegypti, which wasn't really on our radar 5 years ago. Now it's on our radar. It would be kind of a bummer if the cargo that we had blocked other diseases, but not that. And so you'd like to be able to go in and do multiple cycles. You'd also like to get to Andreas's point, you'd like to be able to go in and remove old stuff. Just as a general, just general good house cleaning, genomic house cleaning, you'd like to remove clutter from the genome. And so more specifically, what you want to be able to do is to take a transgenic, a first generation transgenic N, where you spread it into a population, and now you want to put in a new transgenic, N+1, and you want that one to be able to spread into the population of N such that it goes to fixation and at the same time drives out the other element, right? And in that way you've achieved two goals. You've brought in new content and you've overwritten and eliminated old content. So let's look at how you can do this, focusing on a context in which you have a cargo that has failed in some way. So the idea is going to be that we can take advantage of the fact that these cleaver elements are chromosomally localized. And what that means is that if we have an element N here, we can, at the same position in the genome, we can introduce a new element that has new Cas9 and guide RNAs targeting a different essential gene and a new rescue targeting that different essential gene. And then very importantly, also the old rescue that targets N, right? And now we have a situation in which, because these are at the same position, meiosis can't recombine them onto the same chromosome. And so they're forced to compete with each other for transmission to viable progeny. And then that leads to this sort of situation, which is analogous to what I showed you at the very beginning, where those progeny that inherit the cleaver N+1 survive because they inherit rescue N+1, whereas those that inherit only cleaver N die because while they have rescue N, they have no rescue N+1 activity. All right? So very simple logic that allows you to drive something new into a population. So we've actually done this in a couple of ways now, and this is really just the final data slide, where the idea is that we have wild types here, and we have a new cleaver that we call cleaver dribble plus rescue TKO. And so this is a dribble cleaver, and then it has the rescue transgene from TKO. And just as a reality check, we show that this can drive into a wild type population, converting wild type to cleaver dribble TKO. Those are the red lines here. And then we do the critical experiment, which is we take cleaver TKO and cleaver dribble TKO, and we drive it into a cleaver TKO population. So this is a population that, remember, has gone for 20, 30 generations in a completely locked-in state. And what you see is that, again, we can drive this new element into this population. And when we measure the allele frequencies, of course, this new element is at a very high frequency, and the other element is at a low frequency, around 10%. And again, that will go down over time, just as more killing goes on. The controls in these experiments are just taking, say, cleaver dribble and driving dribble TKO and driving it into cleaver dribble, in which case nothing should happen because it's just dribble driving into dribble. Okay. All right, so the summary then is, and I kind of went fast here, so I skipped over the meaning of the whole word resilience. And so what do I mean by resilience? What I mean by resilience is when we think about robustness, we're thinking about the ability of a system to avoid failure by multimerizing components, engineering redundancy. But the other thing that's as or more critical is this idea of resilience, the idea of being able to recover from fact that when you do fail. And so what this system allows you to do is to recover from the inevitable failures that must happen in genetically diverse populations. And again, it involves these incredibly simple two-component system, just a recoded gene and CRISPR and Cas9. And then we can overwrite old content. And this, again, we think is useful because it provides an important point of control. It provides a kind of reversibility. It's not really reversibility to wild type, but it allows you to change a modification that you have made and put something new in its place. And then if you were targeting a haplolethal gene or a haploessential gene, you would be able to use other approaches to actually completely get rid of the transgene in the population by, say, adding wild types to the population so that you fall below that threshold that's needed in order for drive to occur in any case. So let me stop there.